Detection and quantitation of fermentation products, that is the measurement or assay of fermentation products. Assay means measurement of desirable fermentation product from the given mixture of compound, or it may be a pure preparation. These assay procedures must be quick, simple, reliable, and accurate, and they must measure only the compound of our interest from the mixture of several compounds that may be present in fermentation broth. So, this is the criteria while choosing a particular method for measurement of quantity of a particular fermentation product. There are certain general methods for detection and assay of fermentation products which are classified like this. This is the general chart which shows that physicochemical methods such as these physicochemical methods are basically useful for the quantitation of products like organic acids like citric acid, lactic acid, acetic acid, etc. And the method which is used for their measurement are titration and gravimetric analysis. Secondly, the measurement of biomass. So for the measurement of biomass, the general method used for their quantitation is packed cell volume method, cell count method by using standard plate count or direct plate count or direct microscopic count, or the use of different counting chambers like Newborn's chamber. Another method of measuring biomass is turbidity analysis by means of measurement of optical density. Third category of products, that is antibiotics, vitamins, enzymes, carbohydrates, proteins, amino acids, lipids, and nucleic acids. So generally, these compounds are measured with the help of spectrophotometric assay. Amino acids, peptides, sugars, microbial toxins, pigments, organic acids, growth factors such as indole acetic acid or vitamin B12. For such measurements, the common techniques are paper and thin layer chromatography, gas liquid chromatography, high performance liquid chromatography, that is HPLC, and mass spectroscopy. Then, in the category of biological methods, the compounds which can be quantitated using biological methods are either growth inhibitory substances or growth stimulatory substances. That means these methods are useful for the quantitation of antibiotics and for the quantitation of substances like vitamins or other growth factors. The methods which are used for their quantitation are diffusion assay. There are two modifications of diffusion assay: disk diffusion assay or well diffusion assay. Then turbidimetric analysis, that is tube dilution assay, endpoint determination assay, and enzymatic assay. Let us learn about the physical chemical assays used for the quantitation or the measurement of fermentation products in the given preparation. The actual choice of the particular assay to be used It depends on physicochemical characteristics of the product. So these physicochemical characteristics of the product make it possible to use these different methods of assays. First example of physicochemical assays is titration and gravimetric analysis. Generally, it is used for the quantitation of the products like organic acids. So their quantity, it can be roughly measured by pH indicator dye, that is, for example, addition of bromothiamyl blue in the given preparation, and then the end point titration is performed with the help of standard alkaline solution of known strength. So this is a general titration that gives us the idea about total tritable acidity of organic acids in the given preparation. Organic acids may be precipitated and converted into insoluble form. Say for example, lactic acid. So lactic acid, if it is allowed to react with calcium carbonate, if calcium carbonate is added to the preparation containing lactic acid, then it will cause the formation of salt of acid, that is calcium lactate. Then this calcium lactate is washed, it is separated by centrifugation or filtration, it is dried, it is weighed, and then its quantity is measured. So this precipitation, washing, drying, and finally the quantity is determined in terms of its weight. This is called as gravimetric analysis. Second method is turbidity analysis and cell yield determinations. So for turbidity analysis and cell yield determinations, the method used is pack cell volume method, that is PCV method. The tool which is used for this method is a graduated tube, that is PCV tube. This graduated tube, it has marks, as you can see on this centrifuge tube. There are marks which are measured in terms of centimeter cube. Centimeter cube is equivalent to ml. Centimeter cube, it is a measure of volume. So whatever the preparation is there, whose turbidity we want to measure or whose biomass we want to measure, it is poured into this tube. Then this tube is allowed to centrifuge, and finally the mass of microbial biomass which is gathered at the bottom, it can be measured in terms of centimeter cube. That is the method of packed cell volume. Another method is of measuring biomass is turbidity analysis. This is the protocol for turbidity analysis. We'll see it one by one. So the cells are first suspended in the growth medium. So these are the cells which are considered as standard cells. So these cells are first suspended in growth medium. It will serve as a standard reference. Then this suspension is then diluted to a turbidity range, quantitatively measured as optical density. So the first thing is that we must have the desirable organism, we have to take its growth, prepare the suspension and dilute the suspension so that we'll get different ranges of turbidity, different tubes containing different turbidity, different optical density. Then this optical density, it can be measured by using a simple colorimeter, which makes use of a visible range of light. After the measurement of this optical density, in between, to eliminate the absorption by growth medium, because what we are measuring is optical density, which is related to absorbance of visible light from visible range of light. So this light, it can be absorbed by microorganisms as well as it can be absorbed by the components present in growth medium. But we are only interested in absorbance of light by microorganisms, and we are not interested in the absorbance of light by medium components. So to exclude or to eliminate the absorption by growth medium, there is use of a control. That control is uninoculated growth medium. So when you use uninoculated growth medium to set 100% light transmission, so this will help us to eliminate the absorbance by growth medium. Then the turbidity measurements are then standardized against plate counts. That means the first step is you have the organism, suspend the growth in uh, saline or growth medium, and then prepare several dilutions, measure the optical density of each dilution. Now you want to standardize this optical density readings. So we can standardize these readings against plate count by determining number of cells. That means if you have 10 raised to minus 1 dilution, so take 0.1 ml of 10 raised to minus 1 dilution, place it onto a nutrient agar plate, say for example, spread the suspension, incubate the plate, and next day you will get the number of colonies. So this is how we determine total viable count. Then next step, so this thing will be repeated for each and every dilution. Now what you have two parameters. One is optical density of that particular dilution, and you have total viable count or the cell number present in that dilution. So you can prepare now a standard curve relating optical density to the plate count. So once this standard graph is prepared, the optical density readings can be easily converted to cell numbers. This is how we prepare a standard graph. Then like this is the standard graph which is prepared as mentioned. So on y-axis it is optical density, and on x-axis it is the cell number, log of cell number. So this is how we prepare a graph, and now this will be called as the standard graph. Now what next? The next thing is that you have your sample, means the sample whose cell number you want to determine. This is your sample, means the fermentation broth in which the microorganisms are present, and you want to measure how many microorganisms are present in this sample. So what we did, first we prepared the standard graph using standard cells. Once the graph is prepared, now we'll pick up this sample, we'll measure the optical density of this sample, and then this reading of optical density it will be then plotted onto this line, which is having a particular linear line, linear part we have to use, and that optical density, if it is correct, it will be then plotted onto this line. That will give you the reading of log of cell number. This is how you can convert optical density readings into cell number. This is how the turbidity analysis is done. Another procedure for measuring the microbial cells or microbial number is plate counting procedures or microscopic counts. For the counting of cells, there may be use of counting chambers. One of the examples of counting chambers is Petrov-Hausser counting chamber, in which a known volume of suspension, it is placed onto this slide, and then the cells are then observed under microscope and the cell number is then counted. In order to make it visualize, or in order to improve this particular assay protocol, there may be addition of fluorescence stains. Now here the major drawback of this type of counting method is that both living cells as well as dead cells of the bacterium or the microorganism, it will be measured or it will be counted because we are observing this slide under microscope. So microscope will not be able to distinguish between dead and live microbial cells. So in order to improve the quality of the test, there is addition of fluorescence stain. For example, acridine orange. So this fluorescent stain, it will be taken only by viable cells and not by non-viable cells or dead cells. So this is how the cells which contain this fluorescent stain will be only considered as viable cells. Then another method in physicochemical assays is spectrophotometric assay. Spectrophotometric assay, as the name indicates, it makes use of an instrument called as spectrophotometer. So this spectrophotometer is useful for the measurement of desirable fermentation products. Now let us understand exactly, and we have already learned about the principle of colorimetry and principle of spectrophotometry. That if you want to recall that, please go back and read Beer and Lambert's law, which says that as the concentration of a compound increases, the optical density it also increases. So you just go back and see what is Beer and Lambert's law. So this is the principle of spectrophotometric analysis. Now let us see if the fermentation product whose quantity you want to measure, if it is a colored compound, for example riboflavin, it is a vitamin, and riboflavin, it has an orange yellow color. So if fermentation product is self colored, then what can be done? If it is self colored, then we must, you see, for any spectrophotometric assay, the first condition is that you must have a standard reference compound. Standard reference compound, that means if you are working on riboflavin fermentation, then what is the standard reference compound? It will be riboflavin in pure form. So this standard reference compound should be available in pure form. What next? This pure riboflavin, that is standard reference compound, it will be diluted by using suitable solvent as it is shown in the figure. So if it is diluted, then it will have different concentrations of riboflavin in different tubes. You can see the first tube is now having 10 micrograms per ml of riboflavin, and the last tube is having 60 micrograms per ml of riboflavin. So these are the 6 dilution I have mentioned here just for the just as an example. We can see here the color differences. The the last tube having high concentration of riboflavin is little dark, whereas a tube, the first tube that is having less concentration of riboflavin, the color is quite fade. So this color differentiation you will find. Now what is the next step? The next step is to select a wavelength of light from visible spectrum of light. So understanding the principle of Beer and Lambert's law, we know that every colored compound have ability to absorb a particular wavelength from the visible spectrum of light. That is the property of colored compound. So the first thing is that having standard reference compound to be used for the selection of a particular wavelength. How will we determine the particular wavelength? The wavelength of light in visible range that is maximally absorbed by riboflavin. That wavelength should be chosen for spectrophotometric assay. So it is a different exercise. So already now we know that for riboflavin a particular wavelength is selected based on earlier study. So that wavelength will be used within visible range of light. So after that, measure the optical density of all these tubes at specific wavelength. What is that specific wavelength? I have already explained you before how to select that particular wavelength. So the wavelength of light which is maximally absorbed by desirable standard reference compound should be selected as a specific wavelength. Now the color is measured in terms of optical density. The care has to be taken that we want only absorbance by this colored compound. We don't want absorbance by medium components. Therefore, in order to eliminate absorbance by medium components, what is the solution? As we have seen in the earlier slide, we have to use uninoculated medium as control and use this uninoculated medium to adjust 100% transmission. This is how you can eliminate the absorbance caused by medium components. After measuring the optical density, now you have two parameters. What are those two parameters? First parameter is concentration of riboflavin, say for example, that is in micrograms per ml. And you have another information now, that is optical density of that particular dilution. So using this particular data, now you can generate a graph. On y-axis, it is optical density, for example, at 550 nanometers. So this is the wavelength which is maximally absorbed by desirable fermentation product, and that is why it is selected here. And the second data that you are having is concentration of standard compound, that is micrograms per ml. So that will be plotted on x-axis. Now, as per the Beer's and Lambert's law, as the intensity of color increases, or as the concentration of colored compound increases, it leads to increase in the absorbance, it leads to increase in the optical density. So as the concentration is increasing, the optical density is increasing. That means this shows the proportionality between concentration of standard compound and optical density. So we can see here in this graph, as the concentration is increasing, the optical density is increasing, and at a particular point you can see that there is a steady state, or it is the curve is getting bent, or it is getting flat. So that part should not be used for further estimations. What part we can use for reading the results is the linear part of the graph, this part, because here this part, it follows Beer and Lambert's law, and the latter part, it does not follow Beer and Lambert's law. So this part is only chosen. This is how now we have prepared a standard graph using standard reference compound. What next? Next thing is you are having your sample, that is fermentation broth. So this fermentation broth sample, it should be appropriately diluted. The next step will be expose this sample to 550 nanometers and determine its optical density. So measure its optical density and plot that onto this graph. Remember that we have to make use of the linear part of the graph only, where the proportionality is seen, and you have to correlate this optical density with the concentration of compound. That means once you know what is the optical density, you can easily calculate what is the approximate concentration of desirable fermentation product in the given preparation. So now you can convert optical density reading into the concentration, that is micrograms per ml. That is how spectrophotometric assays are used to determine the concentration of fermentation product, colored fermentation product. Remember, colored fermentation product in the given preparation. Now all the fermentation products are not colored. There may be the fermentation products which are non-colored. Most of the compounds are non-colored. So what to do? Still spectrophotometric assays are possible. Very interestingly, what can be done, that these non-colored fermentation products can be converted into colored compounds, provided that there is an availability of certain reagents. These reagents are added to the preparation. These reagents will react with fermentation product, and it will result into the production of a colored complex. So this is how non-colored compounds can be converted into colored compounds by means of addition of certain reagents. Once the non-colored compounds are converted to colored compounds. Now it is very easy to measure or to perform spectrophotometric assay as it was shown in the previous slide. So non-colored compound, addition of reagent, it will generate a color, and now you can perform the assay. So the first step in that will be, we must have a standard reference compound which is non-colored. So that standard reference compound, it must be first diluted as we have seen in the previous slide. So first step is dilution, like 10 to 60 micrograms per ml, say for example. Then to this, the reagent is added or chemical compound is added in equal quantity in all these dilution tubes. That will result into formation of color complex, and the color intensity will differ according to the concentration of fermentation product in that particular dilution, like this. So if it is a low concentration, then after addition of that reagent, it will going to produce a fade color, and then the intensity of that color will further increase. So increase intensity, that is increased, that is formation of dark color, we can see here. So this is how you can convert non-color compounds to color compounds. And the next procedure protocol is same like earlier protocol. We have to measure optical density at particular wavelength, specific wavelength. Specific wavelength, how to choose this specific wavelength, we have already learned. In this case, take the fermentation product, that is pure compound, standard reference compound, allow it to react with reagent, and then expose it to different wavelengths, and then check which of the wavelength is showing or it is absorbed maximally in terms of optical density. Higher is the optical density, higher is the absorbance. So this is how you can select that particular wavelength. So now you have this graph, it will be called as a standard graph, optical density versus concentration. Then the next step is same. You have your sample, fermentation broth, in which your non-color compound is there. So you have to dilute that particular sample, and to that then add reagent. So it will turn non-color compound into colored complex, and then you have to measure the optical density of this and plot it onto this graph. Now you can correlate optical density and corresponding concentration. This is how we can determine the concentration. Now if the sample is diluted, then it should be multiplied with dilution factor. If it is 10 times diluted, then the multiplication factor will be 10. If it is 2.5 times diluted, then the multiplication factor will be 2.5, so on and so forth. This is how spectrophotometric assays can be used for quantitation of non-colored fermentation products. Now the next case is, the product is non-colored and suitable reagents are also not available. Then what should be the strategy? Still spectrophotometric assays are possible. How? So the product is non-colored and there are no reagents available that can convert this product into colored compound. Still, some compounds have ability to absorb shorter wavelength light. Shorter wavelength light means ultraviolet light. It is the property of certain compounds. These compounds can absorb ultraviolet light. So this absorbance can be measured by spectrophotometer, as we can measure the absorbance of wavelength within visible light by spectrophotometer. Using same instrument, we can determine absorbance of ultraviolet light by desirable fermentation product. So the protocol is same as we have seen in the earlier slides. You must have a standard reference compound. That standard reference compound is first diluted like this. These are the several dilutions in desirable solvent. Then expose these dilutions to spectrophotometer, in which each dilution will be exposed to specific ultraviolet light wavelength, and determine the amount of UV light absorbed. So in earlier cases, we have determined the amount of visible light absorbed in terms of optical density. Likewise, in spectrophotometer also we can determine the amount of ultraviolet light absorbed. So now you have two parameters ready, that is UV absorption at 330 nanometers. This is just for the sake as an example. And another parameter you have on x-axis, it is concentration of fermentation product. So this is how you have plotted this graph. Once this standard graph is prepared, now you have your sample, fermentation broth sample. Dilute it appropriately and then expose it to the same ultraviolet light. You will get the amount of UV light absorbed. Plot it onto the graph and convert that reading into concentration of fermentation product. This is how the protocol, even if the product is non-colored and if it is having ability of absorbance of UV light, then spectrophotometric assays are possible. These assays are most accurate assays and very simple assays. Next one is the condition in which the compounds, fermentation products, they have a fluorescence property. Certain compounds have fluorescence property. Fluorescence means absorbance of ultraviolet light, means shorter wavelength light, and emission of higher wavelength light, that is visible light, that is called as fluorescence. So certain compounds have this ability of absorbance of UV light and emitting visible light. So such compounds can be measured quantitatively by using spectrophotometric assay. So this compound, standard reference compound, you have this standard reference compound. After exposing it to ultraviolet light, it fluoresces and the amount of fluorescence is measured. So the protocol is same. You have standard reference compound, dilute that standard reference compound and expose it to ultraviolet light in spectrophotometer. So this amount of fluorescence, the values you will get in spectrophotometer. Now you have two parameters, amount of fluorescence and concentration of fermentation product. So obviously the next step will be preparation of a standard graph. On y-axis it is fluorescence in percent at 300 nanometers, say for example, and on x-axis it is concentration of fermentation product. Now the next step is you have your sample ready. Expose that diluted sample to ultraviolet light. Measure the amount of fluorescence. Plot that value onto, insert that value onto this graph so that you can convert that fluorescence reading into concentration of fermentation product. This is how spectrophotometric assays are used for the measurement of fermentation products, which are colored, which are non-colored but have ability to absorb ultraviolet light, which are non-colored but have ability of fluorescence when exposed to ultraviolet light. So these different types of fermentation products having different properties can be quantitated by using spectrophotometric assay.